And István, then please introduce yourself and feel free to mention anything you like. And maybe also one thing that would be like a cultural tip, something that you like, a book, uh, a TV series, a movie, anything. Thanks so much for being here today. I really appreciate like your time to come and your time to prepare and yeah, to present. Thank you.
Thank you very much, Lucas. Thank you for inviting me here. And being the first presenter, I feel absolutely free to do whatever I would like to. But then also, please tell me if I'm talking about too much on science or anything else. So let me share my screen. And yeah, can you see the presentation? Okay, I can't hear you.
Yeah.
Okay, thank you. Okay, so I'm István Zachar from the University of Budapest, ELTE, from Hungary, and I'm a senior researcher at also at the Institute of Evolution, which is part of the Hanren Center for Ecological Research. This is a pretty recent institute that was established 5 years ago, and we are very small, like 20 or 25 people, regulars, but we are trying to do high evolutionary science there. And I'm going to talk about this older topic of mine, which I have published like 7 years ago. It's the farming, the mitochondrial ancestor, which is an exploitative origin theory of eukaryotic origins. Now, before that, uh, since I was asked to give a cultural pointer as well, I'm gonna talk about one of the books here, which I would really like to recommend, and maybe the other one I will present at the end, so to give a frame for the presentation. So I really like this book, King Jesus, from Robert Graves, who was a British literate person, or poet, a writer, and probably many more else. And this is about the story about Jesus Christ, but in a very non-classical way. So he gives a perfectly viable and believable, credible story, background story for Jesus, which is historically correct. And he, as he writes, undertake. So he I undertake to my readers that every important element in my story is based on some tradition, however tenuous, and that I have taken more than ordinary pains to verify my historical background. And he also claims, I mean, the author, that a commentary for each of the concepts, ideas, and facts that he writes in his book, if he would like to comment on them, that would fill up more than 2 or 3 books. So this is a very nice approach, in an unorthodox approach on the origin of Christianity, and I would recommend it to read for anyone, because the language is marvelous as well. So the guy actually wrote books on the Greek mythology as well. So he is a pretty well-established person in mythology. Okay, so back to science. Here is the roadmap that I'm going to talk about, and feel free to ask questions in between. I'm not sure that I will see. Okay, now I can see the Zoom as well. So I'm going to give a very brief introduction on the origin of eukaryotes and how mitochondria relates to it. And I'm going to talk about the farming hypothesis and how we modeled it in our paper. I try to avoid too much math, but some is necessary. And also at the end, I'm going to give you some evidence that perhaps supports the farming hypothesis. I guess that you are all familiar with the major classes of eukaryogenesis, or how you can classify the different theories. One obvious classification is this time related to mitochondrial integration, and whether then the mitochondria came before major eukaryotic inventions or came after or somewhere in between. Accordingly, you can have mitochondria early theories where you would assume that there is an archaeal host which first acquires the mitochondrion, and then either due to the mitochondria or due to other related events, the complexification of the eukaryotic cell started. So these are, for example, the hydrogen and the syntrophy hypothesis, or the more recent entangle, engulf, and enslavement hypothesis. I'm not going to talk about these very much, but it suffices to say now that that these are expecting or assuming that there was some kind of metabolic interaction between the archaeal host and the mitochondrial ancestor, for example, a syntrophic interaction, which enabled them to get close to each other, and somehow one got into the other. It's always a question mark how exactly. You will see that this is a huge question mark. Now, the mitochondrial late theories assume or postulate that there were eukaryotes already, or proto-eukaryotes, without mitochondria originally, and then they acquired the mitochondria after most eukaryotic inventions were established. For example, the nucleus, the Golgi, or cytoskeleton, and whatever. The classical hypothesis for this is the archezoa hypothesis by Tom Cavalier-Smith, which we all know that is not true or cannot be maintained anymore. But it came back in a different disguise as new archaea were recognized in the last 10, 15 years, the Asgards. So it became a trend to actually assume that the host was a somewhat more evolved archaeon, an Asgard one, which could have been one that was phagocytosing, or having other eukaryotic inventions before having a nucleus. And these pre-eukaryotic features already present in the Asgardian host might have been useful to capture the mitochondrion. And one example hypothesis is the phagocytosing archaeon hypothesis or theory, the FAT theory proposed by Martin and Etama, I think. But I'm going to talk about these later, hopefully. The problem with all of these is that none of them have we haven't found any such intermediate stages that these hypotheses are postulating. So we haven't found mitochondriate archaea, or any kind of archaea with an internal endosymbiont. We haven't found eukaryotes that are primarily lacking mitochondria, and we haven't found archaea that are phagocytosing. So that's a conundrum where we cannot distinguish between these theories based on evidence from existing intermediate stages. Now, the main two contenders are the syntrophic theories and the phagotrophic theories, at least for this presentation. I try to keep it simple. Syntrophic theories assume, as I said, that...
István,
Yep.
Is there any theory in which the agency is also given to the guest? Like in the previous cases, like the host is capturing the guest from the environment. Is there any other view in which the guest would actually invade the host?
Yeah, it's a very good question. And there is, and I usually put up a third class here, the parasitic capture or parasitic invasion hypothesis. And but needless to say, there are 60 or 70 or even more hypotheses out there. But this would be a third class where the symbiont is actually actively infecting the host, and that infection might have led to some kind of prolonged coexistence between the two. Well, ultimately, where ultimately the the parasitic symbiont was attenuated or tamed to be more beneficial for the host. Of course, the big question here is, why would it be advantageous for the host to have an infection? Clearly, it's making the host sick, so it's very hard to explain. There are hypotheses to try to go around the corners and make it viable, but I'm not very familiar with them. We are working on one as well in our group. Yeah, but back to these main hypotheses. One expects that the host and symbiont had some kind of metabolic cooperation, for example, a syntrophic interaction where one party produced something that was beneficial for the other, or vice versa, or it could have been mutual, where both parties produce something useful for the other that helps for them, even the partnership. But then it doesn't have to have phagocytosis, but it's very hard then to explain what exactly ensured the prolonged coexistence of the parties and the vertical inheritance, how the symbiont actually got into the host. And yeah, there are these nice theories floating around, which was presented, I think, in 2014 by Buzz Baum, the inside-out theory, which presumes, and this is a more modern interpretation from 2023. So the inside-out theory presumes that the host producing these blebbings or protrusions increased its surface, and ultimately these protrusions not only captured symbiotic partners, but ultimately they fused with themselves to produce an entirely closed system where the nucleus is the remnant or the how should you say, the inheritor, the remainder of whatever the original cell was. Now, of course, no one's seen anything like this so far, so we still don't have examples, but the recent cultivation of the Loki archaea by the Japanese group proved or provided some kind of support for this theory, because these Loki cells really produce branching protrusions. Not that they are fusing with themselves, we don't see that, but it's something that really is not that common among prokaryotes. Now, on the other hand, regarding phagotrophic theories, you don't have to expect or don't have to assume metabolic cooperation, because there is none. So the would-be host, would just simply prey on the symbiotic species. And phagotrophy then is clearly a mechanism that exists and that can allow the inclusion of the partner. And if the partner somehow survives digestion, then you have very easily created vertical inheritance in this lineage. Of course, we still don't have examples for that because we don't see phagotrophic archaea. Now, nevertheless, phagotrophy is a mechanism that was suggested, I don't know, more than 30 or even 50 years ago by many people, and we all know that phagotrophy was the mechanism for probably all plastid acquisition, and there were, I don't know, many, more than 10 probably. And in some cases, you can even see the remnant of the phagosomal membrane around the secondary, tertiary symbionts. So there's a clear trace of phagotrophic inclusion. Of course, you don't see this around the mitochondria or primary plastids, and we don't know whether this is because the phagotrophic membrane just got degraded or whether it was not there at all initially. So there are many examples for phagotrophic inclusion, and we know that phagotrophy can be beneficial. There is a very nice example, which is called the CET, the microcosm. It's including the tetrahymena, ciliate, the microctinium algae, and the coli bacterium. And the ciliate actually eats the bacterium, the coli, So that's its prey, and it preys on it with a phagotrophic mechanism, like all ciliates do that. And as the experimenters and the Nakajima group added algae to the microcosm, it turned out that the host sometimes fed on the algae, but actually the host could not digest the algae. So the algae either died by whatever reasons within the host cell or survived. But if when they survived, they could continue photosynthesizing and even probably replicate within host vacuoles. So you can see that there are algae cells in host vacuoles. And very interestingly, two different strategies evolved within the algae population. One phenotype tried to become more aggregative in the sense that different algae cells stick together so that they form a big enough aggregatum that cannot be digested, cannot be engulfed by the cilia. And the other one was specifying on becoming a better endosymbiont within the host and perhaps even providing sugars for the host. We don't know that, or at least I don't know that. So this is a clear mechanism that an accidental capture of a partner can lead to prolonged coexistence, perhaps even to endosymbiosis among eukaryotes. And that's just one of many. And that's another image of the tetrahymena. Okay, so this led to the idea of the farming hypothesis, which was proposed more than 30 years ago by John Maynard Smith and Szathmáry in the Major Transitions book. Of course, you cannot avoid to mention this book in such a webinar. And what they thought was that either there was a metabolic, mutually beneficial interaction between the parties, or there was it's even written here that the symbiont was a parasite, but then you have to explain why the host would have any kind of a selective advantage, or the proto-metakaryotic host, whatever that is, we don't care about that. So the host cells may have captured the proto-mitochondria and kept them as humans keep pigs, so to exploit them for a controlled for a prolonged time in a controlled way. So it assumes that the host was a phagotroph and it preyed on the symbiont, but then it could set aside some living prey for later digestion. That was the original idea, and I started modeling this in 2016 or 17. So what we did, and I'm going to talk about the model a bit more in detail, what we did was to assume that there are two phenotypes of the same nondescript species. So it could have been a proto-eukaryote with a nucleus or an archaea without a nucleus. It doesn't matter for the model. So we assume that there was a greedy phenotype, that's the wild type, which eats whatever it captures immediately. It doesn't know how to set prey aside. Now, the farmer phenotype can set aside some of the prey in phagosomes. Phagosomes are not modeled themselves, but it doesn't matter. The point is that it has to carry the living prey within its cell membrane, which poses some costs, obviously, because then it has to feed them. We don't assume that the symbiont is a phototroph. It could have been, but there is no indication that the mitochondrial ancestor, the direct ancestor, was a phototroph. So we did not assume anything like that. So it's a costly thing to maintain such a living stock within the cell membrane, and we also assume that the symbionts could reproduce in the host. Now, obviously, if it has a cost, then the host that can farm is in a selective disadvantage. So we have to figure out some kind of indirect benefit for the host, and then we figured out that if we assume that the environment is fluctuating, then there could be good and bad periods. So where good period is one where the resources are always available, and the poor period is one where the host doesn't have resources to feed directly, or the symbiont doesn't have resources, hence they are dying out and the host wouldn't find prey. So either way, there is an external forcing which was necessary, and also we assume that that this leads to a trade-off, because in good times farming or carrying a farm reduces your own growth rate, while in the bad period, the poor period, it may be the only lifeline for you to survive. So this would be the only slide with the equations, and I try to keep it as simple as possible. So we figure that there are these two phenotypes, the farmer and the non-farmer, the greedy phenotype, and then there are the two environments, the resource-rich and the resource-poor environment. In the rich environment, they are growing exponentially, basically, but of course we had to add the carrying capacity to keep dynamics at bay. So ultimately, it leads to the extinction of one, but practically it will never go extinct, but approximates zero very closely. And in the poor environment, we assume that and in the rich environment, it's always the non-farmer, the purple one, that dominates or wins selection, if there is. While in the poor environment, the farmer enjoys a bit of delayed extinction. So the non-farmer, the greedy one, decays exponentially, because there is no growth in the poor period, just death. So it's a very poor environment for them. While the farmer type can actually live on its own farm for a bit more time. Ultimately, the death rate will converge to the same one as for the non-farmer. Now, in this minimal model, we have not modeled exactly or explicitly, the internal farm size. We were not interested in that, and it's very hard to do in an analytical way. But we are working on that right now after 7 years. But for the minimal model, it suffices that the growth rates are different for the good periods. So the farmer always grows faster. Actually, this should be the other way around. So A should be, A is bigger than B. Oh, no, so B should be bigger than A, because B, okay, the figure is, so the equation is fine. I'm just got confused. So A is the non-farmer's growth rate, and it's bigger than B, the farmer's growth rate. Okay, so that's fine. And then there is a total growth that sets the carrying capacity. So these two sets of equations are what we need, and you can calculate the ratio of farmers and non-farmers at the equilibrium. But before that, I just want to show you that how the dynamics unfold in time. So here you have the non-farmer population that starts with a higher initial value, and as poor and rich periods come in sequentially, then you see that it's always for the start phase, it's always the non-farmer that wins. But ultimately, the farmer phenotype can gain the upper hand, and really, without any kind of additional mutation or anything added to the dynamics, it will ultimately win over the non-farmer. So what does it tell us? That costly farming in good times and delayed digestion in poor times is actually a bad hedging strategy, which is adaptive in a fluctuating environment. And this works even if you have random period lengths, so that the gray ones are the poor periods and the white ones are the good periods. And it doesn't matter what their distribution is, because ultimately it's always the farmer that wins. And this is it's a longer run. So what we did to establish or find out how robust the farming behavior is, was to calculate the limit of the ratio of farmers versus or farmers against non-farmers. So sorry, y over x, which is we call z. And well, it doesn't matter how it can be calculated. It's an algebraic thing, but we could not analytically calculate the limit. So what we did was just to iterate it for n successive iterations, 100 or 200 iterations, and after that we just cut off who has a very low ratio. And then what you see here in 3 dimension against relative farmer decay rate, relative farmer growth rate, and relative poor period length, that there is a quite a large domain where the farmers dominate over the greedy phenotype. So wherever there are colored points, it means that not only that the farmer survives, that most of the time it dominates over the greedy phenotype. So it's an ecologically stable solution. So then we did an evolutionary analysis as well, whether farmers can invade a non-farming population. For that, you only have to know that as we introduce new mutants, the mutations change not only the R parameter, which is how delayed your death is in the poor period. So the more you increase the R, the more closer you get to exponential death. But there was a trade-off. So we not only changed the R, but we also had to change the growth rate of the mutants, because if you are a better or on one hand, if you're a better survivor in the poor period, then you have to have a cost in the good period. So you decrease your growth rate in the good period to increase your survival rate in the poor period, or the other ways around. And what you see here is exactly that starting from the upper right corner, where there is a very high growth rate and very small delay. Well, actually, it's a high parameter value, which means a very small delay on survival. So again, if R is large, then you're close to exponential decay. So this is the wild type. And you start from here, and the random evolutionary walk converges to the bottom left corner, where growth rate is practically minimum. So of course you cannot go to zero, but then the delayed decline property, which is R, is the maximum possible, which means that the survival is more easy in the poor period. So again, if you iterate it for many independent simulations, then you'll see that most of the trajectories are converging from the top right corner to the bottom left corner, indicating that this is a very strong attractor. And this would be the farmer phenotype, and the top right would be the non-farmer phenotype. Okay. And what we did, was that I built an individual-based model as well, which produced very close results compared to the analytical model. Now, the individual-based model could actually explicitly represent the internal farm size of each cell, because each cell was explicitly represented. What you see here is that the blue, the farmer density increases with time, and the non-farmer density decreases. Here, the green shows the prey density, which corresponds to poor or rich periods of time. And then we also had different evolutionary traits, the culling rate, or how effectively you harvest your own farm. So how often you massacre one of your farm members, and also the farming rate, how effectively you capture prey from the environment and allocate it to the farm. And in the long run, you see that the same behavior emerges, that the farmer wins over, but it maintains still a large culling rate, which is expected, of course, because the farmer can only survive if it kills the farm it maintains, which is not something that we see nowadays in mitochondria, because mitochondria are almost always or almost never digested by the host, only under stressful conditions, or I don't even know, under pathogenic conditions. So the last thing I did was to allow the simulation, allow the host, to allow the symbiont to evolve another means of providing a benefit for the host, a direct benefit, which would correspond to something like a host induced or something like how should I put it, a metabolic help towards the host. So but it started out as zero because it started out as a cost. So the symbiont originally has a cost value, that's the red curve here, which shows that the cost with time will decrease as the evolution of metabolic health evolves, or as the metabolic health evolves in the symbiont, and the cost decreases, ultimately bounding back to the positive value of benefit, which would correspond to some kind of sugars or ATP or whatever metabolite provided by the symbiont to the host. And here you see only the benefit in a different time scale, different y scale. It's the same as here. It's just that it won't fit on the normal scale. What you see here is that in the upper plot, the culling rate is still high. It's the teal curve, and it's still high, which means that despite that there is a metabolic benefit by the symbiont, it's still better to eat the symbiont than relying on its metabolic help, which again is not something that you see with mitochondria today. But if the benefit has evolved, so the green curve has already above a certain level, and the culling mutation of the culling is slow enough, then it allows the culling rate to actually drop very close to zero, or you can even lose culling, which means that you won't eat your farm. You will rely on the farm to provide metabolic benefit forever. So that would be a starting point for modern mitochondria. Okay, and this was already suggested by Neil Blackstone in 2013, where he provided that protomitochondria might have emitted ATP to stabilize the host to avoid digestion, assuming that there was a host who either wanted to digest the symbiont based on stress factors or due to being a phagotroph a priori. Yep.
So in this case, you are showing a change in the initial shape of the relationship, right? Something that was exploitative kind of become mutualistic, right? And
Exactly.
that is be driven by the evolution of the guest that's become less costly in a certain way.
Yes, it's a very direct way to model this metabolic health, because I just increase the cost, which would otherwise be negative, but I just mirrored it in the positive half plane. But it's not that I have explicitly modeled metabolites or metabolic interaction. It's just that you don't pay costs anymore. You get something out of the interaction. But I want to point out that I wouldn't say that it's a mutually beneficial interaction in the sense how mutual cross-feeding or mutual syntrophy is, because in this case, the host still can eat the symbiont, and the host does not really help the symbiont metabolically in any way. Of course, it pays a cost. There is always a maintenance cost, but it's not like that the symbiont gains anything beneficial from the host that he wouldn't have gained in the previous scenario. So the symbiont doesn't gain anything. it still can be eaten, but of course it's better for the symbiont not being eaten. So that's his benefit, but it's not a direct metabolic cooperation.
In this more explicit model that you tried, did you also have like symbionts in the environment and they are competing with their own inside of the host?
Sorry, so was it a question?
Yeah, yeah. I was wondering, like, you have the symbionts inside of the host and outside of the host, right? In this explicit model. I wonder if they are competing against each other, like in the population, or that was not considered.
That's a good question, but you always have to factor in that once the symbiont goes into the host, then any kind of benefit provided for the symbiont or for the host will be evaluated as the fitness of the group. So now it's the group that is competing with the symbiont and not the internal symbiont with the external symbiont. So that's the multi-level selection scenario that we were exploiting here. And we are working on similar scenarios nowadays, which are again analytic and not individual based, where we actually try to model this kind of explicit metabolic behavior and whether this is sufficient to enable ectosymbiotic association, so not even endosymbiosis, just ectosymbiosis between host and symbiont. And we got good results, and 2 papers I think are already in the pipeline, but I can share the details of the manuscripts afterward, but this would be a long talk if I were going to talk about that as well. Okay. So this last step would be modeling the evolution of how the host or the symbiont evolved these steps. So these A and T proteins that are presently at the membrane of the mitochondria are ensuring that ATP is provided from the symbiont to the host. Now, of course, this was evolved by the host and integrated by the host, but we ignored that for the moment. Oh yes, so I already told you about this. And lastly, I'm going to just very quickly go through the evidence how or what supports an early phagotrophic origin. We all know that the plastids were acquired by phagotrophic means, or very likely almost all of them. And in 2019, it turned out that even a sister to Rhodophyta, so one of the earliest plastidic plants, so a sister to Rhodophyta has a phagotrophic stage, which means that this Rhodophyta, which are sister to Rhodophyta, have a phagotrophic stage, which means that, or it was interpreted by the authors that this is the ancestral state that survived for this long in this minor group. And the similar phagotrophic sisters to basal fungi were found as well. So this suggests that the archaeoplastidic ancestor was phagotrophic initially, and the phagotrophy was lost multiple times independently in Archaeoplastida. Also, though many claim that phagotrophy is a very costly mechanism and it's unlikely that without mitochondria any prokaryote could evolve it, this turned out to be not true on multiple occasions. Once that Monocerco monoides proves that eukaryotes can actually live a completely full life without mitochondria. So it's a eukaryote which is actively moving. It's phagocytosis, large stuff, and it has a flagellum, but it lacks mitochondria, any kind of mitochondria-related organelle, and also it lacks most of the mitochondrial genes. So there is, of course, there is a bit of trace that it once had mitochondria, but clearly mitochondria are not required, or mitochondrial energy express surplus is not required for phagotrophic behavior or the eukaryotic condition. Also, in 2019, this planktomycete was found, which could actually do endocytosis and even capture other bacteria with its endocytotic mechanism. And it's entirely a bacterium. It's entirely run on a bacterial budget. So the structure and the energy budget that's available for prokaryotes is clearly enough to do this kind of behavior. Obviously, this is not related to eukaryotic phagocytosis, but an alternate convergent evolutionary solution. So this all leads to the return of the archezoa, which was an abundant theory by Tom Cavalier-Smith, who supposed or presumed that archezoa were the basal eukaryotes lacking primarily mitochondria. It turns out that they had mitochondria once, so they just lost it. But it doesn't mean so that the theory was refuted, but it doesn't mean that this archezoa niche does not exist. What I mean is that if you plot all the prokaryotes and eukaryotes in this energy and genome size plot, then there is still a niche in the middle, which is the archezoa niche, which would be like a primitive eukaryote or a rather complex prokaryote that could have evolved eukaryotic features without a nucleus or without mitochondria. And this is from our other paper from 2017. And I'm not going to talk about this one, but it's important to mention that in the meantime, we learned a lot about Asgards, that they are actually having an active cytoskeleton, which was already supposed by or theorized by Denis Searcy more than 40 years ago. And it turned out that they not only have cytoskeletal proteins, but they have the same function as in, or likely the very same function as in eukaryotes. And when the first Asgards were cultivated, and here is a artistic impression of Loki, it turned out that they not only have these protrusions, but there are actin cytoskeleton and filaments in these protrusions. We don't really know what their function is, other than providing a structure. But science is going on, so I expect nice findings in the near future. So in conclusion, phagocytosis is costly, but does not require mitochondria, can run on prokaryotic budget, and it has immediate benefits. You not only can have rich food resources or rich protein resources, but you can even stockpile according to the farming hypothesis, or you can start your phagotrophic career by simply eating your partners, especially if you're in a dense biofilm where there is not much else to eat. We also know that phagocytosis can evolve from extracellular digestion, or we presume that or theorize that it can evolve from extracellular digestion, and many archaea have such extracellular enzymes. It provides a trivial mechanism to include the partner, and it could have evolved in the Asgardian host, or perhaps in proto-eukaryotic host if we assume that it already had a nucleus. This is an image from Christian de Duve from 2007, where he theorized about some kind of poor man's phagocytosis, which could have evolved and was not necessarily comparable to eukaryotic phagocytosis, but something more primitive, which could still provide a benefit according to either the farming hypothesis or the social cheating hypothesis or some other means. But we still don't have examples among Asgards on whether they have phagotrophy or heterotrophic behavior at all. So farming, in specifically talking about farming via phagocytosis, we know that it can enable a bed hedging strategy which is beneficial in a fluctuating environment, and it works without assuming any kind of metabolic help. So you don't have to assume that there was initially ATP exchange between the partners at all. So it's a very natural way to assume the mitochondrial integration started, but we haven't found the missing link, neither for farming hypothesis or the syntrophic hypothesis. So with this, I'm going to finish, and I want to thank these guys who were helping me with my work 7 years ago. We are still in contact, of course, and yeah, well, I can only very heartily recommend Umberto Eco's perhaps most well-known book, the Foucault's Pendulum, which is a very nice take on conspiracy theories about about many aspects of or early Christianity and satanic cults and whatever. And I really like Umberto Eco. I think he's comparable to Robert Graves, and I think someone even told that Dan Brown, author of the Da Vinci Code, was the poor man's Umberto Eco for the reader. And with that, I'm going to conclude right now. Thank you very much.
Thanks so much. And now I have to read those books.
Well, let's start by one.
Yeah. And is there any questions? I don't know if anyone want to ask any question.
I can go. Thanks for the talk, by the way, and also the books. Like there's a definite theme to the books that you've suggested. So my question actually, maybe even before the question, I thought it's really nice that you're discussing so much about like what we know about the Asgard Archaea, for example. And actually, there was a preprint from just a couple days ago, maybe. I don't know if you've seen it, that they found an Asgard Archaea with like internal membrane compartments, like it has just intracellular vesicles. And this is
Very good.
quite cool. But my question is, so maybe first, like with this result of R going down to 0, it went down to 0 regardless of like the good and bad environment switching rates, or was that specific, like it goes down to a value that's set by the rate of switching?
You are referring to this image?
Yeah. Like, why doesn't it, if I would think, if one of the environments, good or bad, is much more common than the other, then R could stop at a value that's higher than 0. Like, I don't see why it should go down to 0 all the time.
Yeah, probably you're right. I don't remember whether we examined this in detail. Here you can see that I specifically checked that we used one. So the good period has a length of 5, and the bad period has a length of 15. So 1 to the 3 ratio. But there was a bit of wiggling with the exact length. So that's why there is a sigma providing a Gaussian random number on the length. But I guess that in a very broad parameter range, this is the behavior what we see. But clearly, yes, you can figure out cases where it would not be beneficial to go down to as close to 0 in regard to both parameters. Well, I have to think about this a bit more. I'm not sure I'm gonna be able to give you a better answer than that.
No, I mean, I think it's really cool. I would expect that. So anytime TD is bigger than TG, like R needs a low value of R would be selected for, right?
Mm-hmm.
Like whenever you spend longer in the bad environment, then you want to optimize what's happening in the bad environment. But I think as soon as you switch this, TG bigger than TD, I
Mm-hmm.
would think R, it's okay if R is pretty high,
I
maybe.
guess that would happen. Yes, that's a reasonable assumption.
Okay, cool, thanks.
Although it depends on as well. Well, yeah, well, I can even check this, so perhaps I'm gonna run a simulation and then dig up the code and then send around results if I get to something reasonable.
But was there, as a quick follow-up, was there a reason why you expect the bad duration to be longer? Like somehow empirically?
No, not really, but we assume that this is like a worst case scenario, where the bad. Well, of course, it's a bit of a preference toward the farming behavior. So yeah, it's perfectly reasonable to ask why it was not the other way around. Hmm. Yeah, let me run the simulation and then see what happens.
Yeah, cool. I think they're really cool results. So.
Thank you.
Any more questions? I don't know if not, maybe we can conclude. And thanks so much again, István, for presenting. I really liked your presentation and also your research. It was very inspiring and cool.
Thank you very much.
And
And thank you for inviting me and organizing the whole event.
mhm. And maybe I can share what would be the next presentation. Let's put in here 1 second. Share my screen. So for share screen, maybe you have to unshare yours first.
Ah, sorry.
Let's see. Share. Okay. So can you see my screen?
Mm-hmm.
Okay. So that was the one of today. The next one is going to be on December 4th, and is going to be on the evolution of dependency cohesion in incipient endosymbiosis. And this is going to be the second one of the webinar series. The third one is going to be on December 11th. That's going to be about inducing novel endosymbiosis in plant bacteria, in fungi. And then we will take a stop for this year and restart next year. And I think that's all for today. Thanks so much, everyone, for coming. And I hope to see you at the next one. Have a good day.
Bye.
Bye. Thanks for organizing.